So I didn't expect to be doing this in my bedroom, but today I will be presenting on how the stiffness of an extracellular matrix signals epithelial to mesenchymal transition. So to begin with, the stiffness of an ECM is its physical property to resist an outside applied force. There are various factors that contribute to this, such as its composition. The more fibronectin and collagen present in the matrix, the stiffer it becomes. Also, in a matrix, there are proteins that can chemically cross-link the collagen fibrils. This cross-linked architecture is stiffer in comparison and is able to resist more against an outside force. Then finally, in an abnormal niche, which will be the topic of today, it associates with cell types that we learned about, such as cancer-associated fibroblasts and tumor-associated macrophages. And these cells apply a tension force on the ECM, which increases its stiffness. So I'm going to briefly review what we learned about from class about EMT. So EMT is when epithelial cells lose their cell-to-cell and cell-to extracellular matrix adhesions and adopt a more mesenchymal and motile phenotype. So I just pulled up the original Levento paper imaging that we saw with the amino-stained mammary acini, and I just added the experimental parameters and a quantification to it. So ERBB2 we saw in classes being abnormal. This is an oncogene that Levento et al. Transformed mammary epithelial cells with. So we currently have two types of cells, a control cell line and an oncogenic cell line. Next, ribose was also designated as another abnormal condition, and this is a sugar that they added to the matrices to make it stiffer. So we have two cell lines and two stiffnesses. And then in this final image, we saw this as being metastatic with the acini breaking down and the cells escaping, is when the oncogenic cells were cultured in a stiff ECM. They were very motile, and this can be seen as being very cancerous, which we can see on this invasive assay over here. So when I was reading about EMT, I continually saw breast cells being the cell type of study, and the reason for this is because it's very clinically relevant. Oftentimes, breast cancer is diagnosed with a mammogram, and with this imaging, it's become known that tumorigenesis and metastasis is much more likely to occur in a dense breast than a soft breast. So, the dense breast in terms of our class is a breast with a very stiff ECM, which can be visualized as white. So we see our probably collagen, fibronectin, and other such proteins. And then the black, which is much more apparent in a soft breast, is adipose tissue. But tumorigenesis can still occur in a soft breast, so we see this low white tumor here, but much more readily in a stiff ECM, in the dense breast. So Wei et al. Wanted to elucidate the molecular mechanisms as to why this occurs, why a stiff breast can promote tumorigenesis and metastasis much more readily. And they hypothesized TWIST1 may be playing a role in it. TWIST1 is a transcription factor involved in EMT in embryonic development, so it's a good protein to study and see if it's involved in breast cancer. So they did a similar setup to Levento et al., where they cultured the mammary acini in a stiff gel, but this time they also treated these cells with either a control RNA, which did nothing, or a TWIST1 silencing RNA, which would inhibit TWIST1 activity. So they immunostained for cell-to-cell connections to visualize EMT, and without the RNA, the inhibiting RNA, they saw that EMT occurred at a high stiffness, so we can see all these guys escaping and breaking down the acini. But with the inhibitory RNA, this was pretty much inhibited. The acini isn't as broken down in comparison. The cells aren't mesenchymal and moving away. So this is pretty good evidence that TWIST1 is playing a role in inducing EMT. Next, they wanted to see the upstream factors, what is activating TWIST1 and causing it to induce EMT at a high stiffness. and they hypothesize that integrin sensing is involved since it is sensing these changes in stiffness. So, similar setup, repeats the procedure, but this time they stained for twist 1. When twist 1 is inactive, it's localized in the cytoplasm, but when it's active, as seen in the high stiffness matrix, they translocate it to the nucleus. However, when treated with a integrin blocking antibody, EMT was inhibited in a high stiffness and twist 1 remained in the cytoplasm. So this is good evidence suggesting that integrin signaling using FAK, the focal adhesion kinase, really don't have enough time to talk about it, but they did prove FAK is involved, would activate twist 1 and cause it to translocate into the nucleus, driving EMT in a stiff matrix. However, as I just said, it's not so simple. This is only in a stiff matrix. In a soft breast, often, if a tumor does form, it often has a worse clinical prognosis, which is confusing because we just spent all this time talking about the effects of a stiff breast. So Plunkett et al. Wanted to elucidate the molecular mechanisms as to why a soft breast may induce EMT. And they did a similar setup to Wei et al. And Levento et al. With the soft and the stiff matrices, culturing acini in them, and staining for EMT. But this time, they also cultured a mammary epithelial cell line transformed with a RAS oncogene, which they designated as 10AT. So we have this 10A cell line and a 10AT cell line. And interesting enough, they found that the 10AT cell line was undergoing EMT regardless of cell stiffness, matrix stiffness. When they stained for twist 1 activity, they found that it didn't really there were no differences in translocation between the soft and the stiff matrices, suggesting that it's not integrin signaling that's playing a role in inducing EMT in this RAS cell line. So, since they transformed the epithelial cells with RAS, they checked the RAS pathway, specifically ERK. ERK is a kinase that regulates transcription in the RAS pathway, so they decided to check its activity with a Western blot. And they found that the phosphorylated version, the activated version, was way upregulated in the 10AT cell line compared to the 10A cell line. As we can see with this quantification, their activity is very similar regardless of stiffness, but overall greatly than the normal cell line. They next want to see how ERK was affecting EMT, specifically cytoskeleton reorganization, and found that it acts on microtubule reorganization. They discovered this by treating the 10AT cell lines with either actin inhibiting drugs or microtubule inhibiting drugs, and found that only the microtubule reorganization inhibitors stopped EMT from occurring. So these are more epithelial-like. It's not as the lamellipodia are not as spread out as seen with the actin inhibited cell types compared to the microtubule inhibited cell types. So just to summarize these results, they hypothesized that an oncogenic RAS is activating MEK, which is a kinase in the cascade, which overactivates and phosphorylates ERK, which would then regulate transcription that leads to microtubule reorganization, driving EMT in a soft matrix. So, just to summarize everything, here's everything we've gone over. We see our good friends integrin, FAK, ERK, twist 1, but this is just a sliver of all the current research going on about EMT. We see cytokines, RNA, reactive oxygen species. So much going on, so much I don't have time to talk about, but I just want to show you what's going on in current cancer research. So I know this is a lot to take in, so I'm just going to end it off with my dog Marshmallow. Thank you so much for listening, and yeah.